Cheong Xin Chan, the University of Queensland, Dynamic Genome Evolution of Coral Symbionts, a Bioinformatics Challenge. Hello all, I am Cheong Xin Chan, better known as CX, and I am based at the Australian Centre for Ecogenomics at the School of Chemistry and Molecular Biosciences at the University of Queensland in Brisbane, Australia. First of all, thanks very much to Lenore, Judith, and Holly for giving me this opportunity to share some of our research with you. When we consider a coral holobiont, we consider not only the coral animal, but also the symbiotic dinoflagellate algae known as Symbiodiniaceae and the other associated microbiomes such as the bacteria and viruses. A healthy symbiotic relationship among these organisms contribute to the ecological success of the coral holobiont and therefore the coral reefs. Our interest is in the Symbiodiniaceae dinoflagellates, shown as golden cells here within coral polyps, and they are the most dominant symbionts in corals. Environmental stresses, such as an increase in seawater temperature, would lead to the breakdown of this symbiosis between coral and the algae in a phenomenon known as coral bleaching. This puts the corals at risk of diseases and eventual death if this relationship is not soon reestablished. Symbiodiniaceae represents a diverse family of largely symbiotic dinoflagellates that represent a broad range of ecological niches too. Many are mutualistic symbionts, some are opportunistic symbionts, some have not been found to be associated with any host, so they are considered as free-living. Among the symbiotic ones, some are host generalists, some are more picky and are host specialists, and they also have different transmission modes, vertically from one host generation to another, horizontally between different hosts, or, a bit of both. This slide shows you some examples of where the Symbiodiniaceae can be found. As you can see, they are not only found in diverse corals, they are also found in other animals such as the jellyfish, giant clams, and even the other microorganisms called foraminifera. More importantly, Symbiodiniaceae are a specialized group of dinoflagellates, and dinoflagellates are a vastly diverse group of algae or phytoplankton that also include the bloom-forming toxin-producing species that cause red tides. Although dinoflagellates are ecologically important, little genome data are available for these algae. Why? Because the genomes are highly idiosyncratic. They have enormous genome sizes ranging between 1 to 250 gigabase. That's more than 70 times larger than human genome alone. The chromosomes are permanently condensed in liquid crystalline structure. The genomes are highly methylated. The number of chromosomes remain unclear. However, two recent studies published as bioRxiv preprints in early July this year, just last week, show that some species likely to have 91 to 94 chromosomes. The genes also have non-canonical intrasplice sites, and they have extensive RNA editing, not to mention their organelle genomes are also strange. All these features are against the backdrop of genetic transfer, duplication, and extensive repeat content. In a recent opinion article led by Raul Gonzalez-Page, we put forward a hypothesis of what we would expect from Symbiodiniaceae genomes, given that these algae encompasses a broad spectrum of facultativeness in their symbiotic associations with diverse hosts. For instance, we expect the free-living species to have a larger genome size compared to the obligate symbionts, who have smaller population size, thus have more restricted access to novel genetic material. We will also expect the genomes of facultative symbionts that would be, in and out of the host, to show greater extent of structural rearrangements than the obligate symbionts. So far, we have generated genome data from 10 isolates of Symbiodiniaceae and their 2 free-living relatives, with genome sizes range between 0.7 gigabase pair to 3 gigabase pair. This table shows the available genome data to date. Those highlighted in yellow were generated by our team. We are also involved in the genome sequencing of those highlighted in orange, which are currently in progress. So far, we have been using a combination of Illumina and PacBio technologies in generating these data, and our assembly approach largely follows industrial standard. If you'd like to find out more, please reach out via email. The more time-consuming and the more challenging step for us is actually to develop a customized workflow for ab initio gene prediction from dinoflagellate genomes, given the limited dinoflagellate gene and genome data available at the time and their atypical gene gene features. We, especially Tim Stevens, Raul Gonzalez-Page, and Yi Chen, spent a lot of time on this. And we ended up with an approach that integrates support evidence from transcriptome and protein sequences to yield a high confidence training set for gene prediction. And then we use a meta-method approach to incorporate prediction results from multiple methods to yield the high quality gene models at the end. This process also involves a two-phase strategy for removing putative contaminant sequences. We compare the published genes that were predicted using different approaches, we call them the alpha genes, as shown in orange here, against the genes that were predicted consistently using our approach, or we call them the beta genes in purple. We did a principal component analysis based on the gene matrix from these two data sets. And as shown in this graph, compared to the published genes, the beta genes show a smaller range of variance related to methodological biases across the x-axis, but they show a greater variance related to phylogenetic relationship on the y-axis. So the separation of the different genera is actually greater for the beta genes than the alpha genes. This suggests that a consistent gene prediction workflow yielded genes that correlate better to phylogenetic relationships of the distinct taxa compared to the previously published genes. More importantly, this difference also contributes to significant bias in the downstream analysis of homologous genes and comparative genomics. As shown in this example highlighted by the red arrows, the number of conserved gene blocks among the 5 taxa here and here is quite different between the two scenarios of the alpha versus beta genes. As you can see here, we have about 40 blocks implicating close to 200 genes, compared to about 20 blocks implicating less than 50 genes. They are quite different. This observation highlights the fact that a consistent gene prediction workflow is important for comparative genomics of dinoflagellates, such that the genes should be comparable. Now, let's have a look at how the different symbiotic C genomes compare against each other. This is largely the work of Raul Gonzalez-Page for his PhD project. This figure shows pairwise whole genome sequence similarity among the distinct symbiotic C genomes. The larger and darker the square for each genome pair, the more similar the genome sequences are. The smaller and lighter the square, the more different the genome sequences are. The taxa shown in this square box here represents those from a single genus, the Symbiodinium. As you can see, for the 3 isolates of the same species, S. microadriaticum, they have very big boxes, which means that they have very big squares, which means that they are very, very similar to each other, which is not surprising because they are the same species. But what we can see here among the species within Symbiodinium genus, though, is that the sequence divergence among these species are very high, such that it is comparable between the different genera, as you can see outside of this square box. This divergence also follows the specialization of the distinct species in their ecological niches. For instance, S. natans and S. pilosum here are free-living taxa compared to the other Symbiodinium taxa that are symbiotic. It just shows how remarkably divergent all the different genomes of Symbiodiniaceae are. Similarly, when we look at the conserved gene blocks among the different genomes, we observe a high extent of structural rearrangements even among the same species, as shown for these 3 S. microadriaticum isolates here. This observation suggests that structural rearrangement is a contributing factor to high genome divergence of Symbiodiniaceae. So not just sequence identity, but also structural rearrangement. This pattern is consistent with the phylogenetic signal we captured using an alignment-free method based on the shared k-mers between each genome pair. Here, k-mers are simply short genome subsequences at a defined length of k. As shown in this phylogenetic network and the phylogenetic tree on the right, the Symbiodinium taxa can be resolved into 4 distinct clades, suggesting potential 4 subgenera within this genus alone. This result is also supported by our analysis of homologous gene families, which I won't have time to go into today. And overall, the results provide novel insight from the conservation viewpoint that one-size-fits-all approach with Symbiodinium C for designing environmentally robust corals may not be effective. Now, when we compare the Symbiodinium genomes, we found that the genome size for the free-living species is smaller than that of a symbiotic species, as shown here. The larger genome size of the symbiotic species appears to be contributed by extensive gene identification events and the abundance of transposable elements, as shown by the red arrows on this graph. However, we did not observe a significant expansion of gene families in the symbiotic genome, of the symbiotic species, suggesting that most of these duplicated genes likely underwent divergence and functional innovation such that they become remote homologs. Now, sex is postulated to occur in Symbiodinium C. It enhances efficiency for selection, but it has not been directly observed. So our PhD student, Sarah Shaw, in a comparative analysis of genomes and transcriptomes, found a complete absence of genes in Symbiodinium C that are involved in the formation of the synaptonemal complex, although other genes relevant to meiosis are present. The synaptonemal complex found in most eukaryotes mediates homologous chromosomal pairing and class 1 crossovers. Our results suggest that Symbiodinium C adopts a synaptonemal complex independent class 2 crossover pathway instead. We hypothesize that this is due to, at least in part, the permanently condensed chromosomes and repeat sequences in Symbiodinium C. In addition to Symbiodinium C, we also sequenced the genomes of the free-living species sister to this family. Polarella glacialis is a cold-adapted dinoflagellate species found only in the polar regions. The genomes provide an excellent outgroup for studying Symbiodinium C and a great dataset to investigate how these algae thrive in extreme environments. This is largely Tim Stevens' work for his PhD project. We generated 2 draft genomes from Polarella, one from the Antarctica and another from the Arctic. Interestingly, these assemblies appear to be of a diploid genome for each isolate. Perhaps more importantly, the 2 genomes have different sizes. The genome of the Antarctic isolate has about 360 megabase more than the genome of the Arctic isolate. And this difference does not contribute to the difference in the number of genes, but we found that this region is really all repetitive regions. So it's either an expansion or the constriction of repeats in either one of these genomes. In fact, about 68% of Polarella genome represents repetitive elements. When we compare the Polarella genomes against the Symbiodinium C genomes, we observe a clear trend of genes in these genomes that tend to be encoded in unidirectional clusters, as shown in this figure here, showing the number of gene orientation change within 10 gene windows across the genome sequences. This trend is now observed in the relatives of dinoflagellates, as shown for Tetrahymena and Plasmodium here. About a quarter of the genes, of Polarella genes, are comprised of single exons. Many of these encode for critical functions. They are not bacterial contaminants, as they are supported by PacBio-ISO-seq full-length transcript data for which eukaryote genes were selected. Now, many of these single exon genes occur in tandem repeats, just like the ice binding domain encoding genes shown on this slide. ISE-binding domains are found in various co-adapted eukaryotes, and they are known to have a reasonably horizontal gene transfer from a bacterial source. Using phylogenetics, we confirmed that the ISE-binding domains in dinoflagellates have a single evolutionary origin here from a bacterial source. This same trend is not observed in other eukaryotes, as ISE-binding domains in other co-adapted microbes, such as the ice diatoms and ice fungi, appear to have multiple origins. Overall, our results demonstrate how selection acts within the context of a complex genome structure to facilitate local adaptation. It appears that Polarella glacialis has utilized a variety of strategies to enhance transcriptional response, including unidirectional coding and tandem duplication of single exon genes that encode functions critical to survival in polar environments. We are starting to see some of these characteristics in symbiotic C genomes as well, but to a lesser extent. And we would love to explore this further as more and more genomes become available. Hopefully, I have convinced you today that genomes of coral symbionts are weird and fascinating at the same time. The more we study them, the more we find new things to explore. The recent achievement of chromosome-level genome assembly of one of the species using high-C technology is no easy feat, and I'm excited to see how the research in this space progresses. Ideally, rounding back to corals and coral holobionts, we want to adopt a multi-omic approach, integrating genome, transcriptome, proteome, metabolome data to understand how the different biotic components contribute to a healthy, sustainable coral holobiont. Now, this slide shows a figure from our recently published work of a coral hologenome that shows how the different components contribute to sustain this holobiont. And this work took many years of coordinated effort and substantial amount of funding through large international consortium. And these are all mainly based on genome data alone. However, hopefully with the advancement of genomic and bioinformatic technologies, we can do this in a greater extent with less money and time. This is our team at the Australian Centre for Ecogenomics, or ACE at UQ. These are the people who did all the hard work. We also recently welcomed Catherine Dugan from Florida to our team as a new postdoc. She arrived in time for the COVID lockdown, but there will be more genome research work in symbiotic C space, particularly for Cladocopium, Durustinium, and Afrinium, so watch this space. Before I stop, I would like to thank all our collaborators and funding agencies without which our research would not be possible. Most of the research I presented today are funded by the Australian Research Council, and the hologenome work was a keystone project of the ReFutures Genomics 2020 Consortium, championed by the Great Barrier Reef Foundation. If you have any questions, feel free to reach out to me. Thank you.